This is episode 238 of the Stem Cell Podcast, Skeletal Stem Cells, with Dr. Noriaki Ono. Hey, everybody, we are Daylon James and Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Stem Cell Podcast, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Noriaki Ono from the University of Texas on the podcast to talk about his research on the function of skeletal stem cells in development, disease, and regeneration of bone and cartilage. We've also got our usual roundup of recent highlights and stem cell news. That's coming right up.
But first, are you attending ISCCR 2023? Daylon, myself, and the entire Stem Cell Podcast team will be attending the meeting in Boston, Massachusetts from June 14th to the 17th. If you're attending the meeting, drop by the exhibitor hall to find the Stem Cell Podcast booth and learn how you can be featured on a future episode. We'll see you there.
All right, Arun, I'm kicking off the roundup with a bombshell. Very exciting for the world, also for my specific field. This is news that came out of the International Summit of Human Genome Editing, the third international summit. The second international summit on human genome editing took place just over 4 years ago, November 2018. You know what they announced then? Yeah, John Hay doing genetic engineering of human babies, right? So that was another bombshell. And here at the very next international summit, although it took some time, they have another major announcement. In this case, I'm not going to bury the lead here. It's generation of live offspring from 2 males in mice, okay? This is a story from one of my idols, Katsuhiko Hayashi, who came out of Mitanori Saito's lab and has been made famous now for his work on generating germ cells from pluripotent stem cells. And in this case, he really went above and beyond. This is work that he's been moving towards for over a decade, and he's finally accomplished it. Much congratulations to him. And that's it. I mean, the story is he was able to make an XY background into XX and generate primordial germ cells and then oocytes from that background. And I think what's interesting here is the way he went about it, as opposed to the way that maybe a lot of people thought it might be done. So primordial germ cells, which are the precursors of sperm and oocytes, they arise in the extraembryonic region and then they migrate to the genital ridges. And that's where sex differentiation occurs in response to these local factors that are present in that somatic environment. And for that reason, the primordial germ cells are thought to be bipotent. Even XY could become an egg, given the right environment, given a female gonadic somatic environment. But despite that idea and this bipotential capacity for the primordial germ cells, many studies have shown that when you try to do that, when you try and differentiate a XX primordial germ cell into spermatogonia, it doesn't work at all. If you try and generate XY primordial germ cells into oocytes, it does work, but they have very low fertility. And that's starting from primordial germ cells that arise in mice in these cases. And this has been attributed to the fact that in these XY chromosomes, you have mispairing of a heterogeneous sex chromosome, so the XY mispair in trying to make an oocyte, or you get ectopic expression of these Y-specific factors because the Y chromosome is there. So the first part of this study was just to confirm that. They showed that either if you start with XY or if you get dropout in just like a XO, a non-XX background, that you really can't get primordial germ cells to generate oocytes. And this is where they, went into the serious engineering part of this story to get to the endpoint, which was this bipaternal offspring, is they just, broke the thing down to the guts. They took the XY background and had to remove the Y chromosome and then duplicate the X chromosome. And this is predicated on the idea or the fact that's been observed that 1 to 3% of XY pluripotent stem cells, they spontaneously lose their Y chromosome. So you get 1 to 3% of these XY background, they'll just spontaneously become XO, or just a single X chromosome background. And so the group, Hayashi's group, they just cultured ES cells for 8 passages and found that a small percentage of them lost their Y chromosome. In this case, 6%, 5 out of 87 subclones. And then in those subclones, they were able to duplicate the X chromosome because, and this is getting a little bit freaky here, uniparental disomy of the X chromosome, it happens. X chromosomes undergo this uneven distribution into daughter cells, and they can be duplicated. Although that's quite a rare occurrence, the Hayashi group used a couple methods there to enrich for that. One was they knocked in this DS red into X into the X chromosome so that they could gauge by way of fluorescence intensity, 2X chromosomes, 2X the intensity, they could identify which of those subsets underwent the duplication. So that was the readout. And then they enriched for this misdistribution by using reversing, which is an inhibitor of spindle assembly checkpoint that enhances the missegregation. And that was it. I mean, they were able to then duplicate the X chromosome, and then they went through the protocols that Hayashi group has established over many years to differentiate first into primordial germ cells, and then using ovarian somatic cells to differentiate those into functional oocytes that then gave rise to live birth. And I think an important note here is that you may be wondering, with all these chromosomal manipulations, what does that leave for a starting point in terms of the genomic integrity? And they did actually do DNA sequencing analysis of the ESL clones that underwent this Y dropout and then X duplication, and showed that they are, 1, euploid, 2, didn't have any large insertions or deletions that were detectable at the resolution used for this analysis. So I wouldn't say ironclad in terms of whether or not there's some genetic aberrations there at the single nucleotide level, but I mean, forget that. This is a huge result, and I don't know that it's going to change clinical care. Arun, I've been outspoken about whether or not it's ever going to be safe to use gametes that are derived from embryonic stem cells, pluripotent stem cells in the human. But regardless, I mean, this is an amazing feat that I think really boggles the mind and borders on science fiction, and the day has finally arrived. Congratulations to you, Dr. Hayashi.
Yeah, and you and I are both science fiction fans. It's been noted here on the show before, and I agree with you. This is one of those sci-fi becomes sci-fact moments in the stem cell field. And I definitely think that in this subfield of stem cell biology, which is reproductive biology, there have been a number of these bombshell stories over the last couple of years. We've talked about Magdalena Zernicka-Goetz, and also, Jacob Hanna's, their work on the roller cultures and the artificial embryo work, synthetic embryos. And now here we are generating viable functional oocytes from male mice, something that I think, and you're closer to the field than I am, I think this was inevitable. This is something that was going to happen in the next few years, but here we are. Here it's it's very exciting to see this happen in mice. And of course, inevitably people are going to ask, and certainly people are asking, given the prominence of the study, when is this going to be possible in humans? And I was reading some of the press pieces that came with this article, and I believe the Hayashi group mentioned somewhere that they're spitballing, ballparking a little bit here, but they're saying that within 10 years, they will be able to make this happen in humans as well. As we know, human reproductive biology is extremely complicated, much more complicated than these mouse studies. And there are a lot of experimental and technical details and hurdles that have to be overcome, but it's the hope that gets you, right? And certainly there's a lot of hope for gay couples in particular with this type of technology. That's really one of the major pieces of hope with this particular study. A couple other things just to allude to and to emphasize: the percentages were low in terms of the efficiency of the process. There was a lot of engineering, as you also mentioned, that had to happen, and multiple steps along that process, the percentages were low. So that in itself just is reflection of how impressive this feat is and how impressive the Hayashi group has been in making this a reality. One other science fiction thought out there is perhaps you can, I don't know, maybe this is something they're considering doing, and I don't know what the downstream ramifications of this possibility would be, but what if you could self-fertilize? I don't know, make an oocyte from a male mouse and then self-fertilize it with that male mouse's own sperm. Is that viable? Why you would do that? I don't know, other than scientific curiosity, but maybe that's something that they're considering here. I don't know. The Pandora's box is open here, right? I mean, this is an incredible study, one of those really landmark papers in the stem cell field, and I'm very excited to cover it here today.
Yeah, man, you said science fiction to science fact. And that example or possibility that you're alluding to, I think that's totally in the ballpark. And I guarantee you there's some people like maybe Jeffrey Bezos out there trying to throw a few $1 million to fund a little kind of pseudo clone of himself. But I want to emphasize again, I mean, you alluded to Hayashi group, or you specified a Hayashi group talking about 10 years. But I think Hayashi was also very careful to emphasize that was only provided that the safety is very well established. And while the practicality and efficiency is a sticking point, I think the major sticking point here is all the genetic manipulations that have to be undergone in order to reach your endpoint. In spite of the fact that these genomes look normal, I think the risk is too high, and the resolution of our analysis is not fine enough to really definitively demonstrate that a cell gamete at this point is not going to be introducing some germline modifications that are irreversible and might irrevocably alter our society and our biology. So I think while 10 years may be practical, I think it may be a long time before we're ready to actually practice this. But that doesn't stop our phones here at the IVF clinic from ringing off the hook from gay couples or just, interested couples who want to know what the potential for this technology is in humans and clinical translation. And I'm not the one to answer that question, thankfully, but I will say it to you, as I've said before, Arun, I am very skeptical about the safety and concerned given the stakes.
Absolutely. And I completely agree with your skepticism. And there is certainly, after what happened with CRISPR, in the human embryo editing a couple of years ago, I think the entire field is on alert and is aware of the ethical dilemmas that are associated with some of these technologies, especially anytime you're messing with reproduction, right? Whether it's, genome editing of the early embryo, whether it's generation of sperm and gametes, anytime you're touching that topic of reproduction, it's going to be certainly ethically charged. And actually, kind of on that note, and hinting at a future episode of ours, we are going to be having a conversation with Dr. Insoo Hyun in a few weeks here, who's actually a prominent ethicist in Boston, as one of the leaders at the ISSCR, and can't wait to talk to him about some of these really exciting embryo technologies that have come about in the last couple of years. So stay tuned for that. Moving on to a Nature Genetics paper. It's certainly not that level of a bombshell, but it's very exciting in my mind, and it's shedding light on a phenomenon in liver regeneration that I'm not super well versed with, and I actually had no idea that this particular concept was a reality, of bipotent transitional liver progenitor cells contributing to liver regeneration. That's the title of the story. Apparently, after there's a really severe liver injury, and hepatocyte-mediated regeneration is impaired, as we know, the liver is really regenerative, right? It's that hallmark, that quintessential regenerative organ in the human body. In really severe cases of injury, those regenerative properties in the hepatocytes are nullified. Okay, they're lost. But what I didn't know, and again, I'm not a liver biologist, maybe this is a common fact for liver biologists, but I didn't know that there's other cell types like biliary epithelial cells, or BECs, that can actually transdifferentiate into functional hepatocytes in cases of injury. So this phenomenon, this transdifferentiation, is certainly very exciting, but it hasn't been fleshed out. Okay. And the subset of these biliary epithelial cells that can, have this stem cell potential and transforming into hepatocytes, that needs to be fleshed out. Okay, that those subsets need to be identified, characterized, and also the mechanisms actually enabling this transdifferentiation process have to be figured out, right? So what they did here, and this is the lab of Bin Zhou over in the Shanghai Institute of Biochemistry and Cell Biology, they identified a transitional liver progenitor cell, or TLPC as they called it, which actually originates from these biliary epithelial cells and can differentiate into hepatocytes during regeneration from severe liver injury. They did a bunch of lineage tracing here, dual lineage tracing approach. I think that's probably why this is a Nature genetics paper. We don't always cover a lot of Nature genetics paper here on the show, but they did a ton of genetic lineage tracing here, using Cree-ER as their driver approach. And they found that these TLPCs, or transitional liver progenitor cells, are bipotent. So they can either turn into hepatocytes or they can actually readopt a biliary epithelial cell fate and phenotype. And mechanistically, what's the driver of this whole process? Well, it's Notch and Wnt beta-catenin signaling. Maybe not super surprising. That can actually orchestrate this bipotency, and whether these bipotent these biliary epithelial cells can transition into hepatocytes. Okay, so that signaling pathway is critical for this transition process. And it's really neat. I think it's, relatively straightforward study, a lot of nitty-gritty lineage tracing that had to go on to make this study a reality. But I think it's shedding light on a phenomenon that I had very little information about. But I think it's falling in line with some of the other stories that we've talked about recently on the show of transdifferentiation, and how this process whereby cells can directly transform from one cell type to another, it might be more common than we'd like to think in the human body. So a neat story, really nuts to bolts about this transdifferentiation process. And I'm excited to see what other stories are coming out that unveil that transdifferentiation is happening all the time in our bodies, like from head to toe.
Yeah, and if anyone's gonna put their finger on it, it's Bin Zhou. He's been a total boss when it comes to these reporter lines and Cree models and locks and all that. He for years. I remember I was in a session with him where he went over all the Cree mice that he had underway, and there was literally like 25 plus at different stages of development. And I'm really gratified to see how he's applied them. A tool maker, tinker, now really getting at the underpinnings of biology. And in this case, it was always about cell fate with him in different organ systems and transition. But here, I love this idea, as you said there, these transdifferentiation events, as we've grown accustomed to considering all these systems in the static steady state. More and more we're appreciating the flux between cell types, and this is a system that I think very elegantly illustrates that flux. And as you said there as well, I think it's just the tip of the iceberg in terms of transition and plasticity in our body in many different organs. So another great study from Bin the boss, and I'm just looking for his next Cree line. I mean, he's also been very generous providing them to the scientific community. So a real contributor in many different facets, and a great story here.
Yeah, that's great to hear. I mean, anytime a prominent scientist can freely and openly provide their tool sets to the greater scientific community, like, I'm a fan of that person, whoever it is, whether it's, people depositing their plasmids on Agene, or sharing their cell lines, or sharing the Cree mice. That's how ideally science should be conducted, right? I think this is a really cool story, and I'm going to get my stem cell scientist card revoked here in a second by saying what I'm about to say. I'm going to say that pluripotency is overrated. Okay, so we don't need pluripotency when you're talking about cell fate transition. You can go straight from one cell type to another in this transdifferentiation approach, and people have artificially done that over the years. I remember one of the first papers that got me really excited about cardiac stem cell biology was actually this paper from from Deepak Srivastava's group over at UCSF, like about 10, 15 years ago, where they directly converted fibroblasts into functional cardiomyocytes. And I was like, whoa, that's just a concept that was so foreign to me. I thought you had to go through the stem cell state. Now, the efficiencies were low back then. They've improved over time. But the other exciting part of a story like this is that transdifferentiation idea it's not just exogenous. It's not something that's artificial. It's something that's happening daily in the human body in cases of injury, such as what's happening in the liver. And I think that's the next step to figure out where else is this phenomenon happening, and what other context of, just homeostasis or injury. I'm just excited about the concept. All right.
Excited. I'm a little bit perturbed here. It's blasphemy. First, you're loving mice. I thought you were a cell guy, and now you're talking about pluripotency's overrated. Oh my God, do you have a fever, Arun? What's going on? But I agree with you 100%. Plasticity is underrated, and I think there's a lot of precedent in our body, as we said. So a lot more to come, hopefully, in the field of plasticity. And who cares about pluripotency, right? Ha 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 ha. Well, I'll tell you, somebody who cares about pluripotency is using it to great ends. And I have to apologize in advance. You probably think I'm playing favorites here, going with my neighborhood girl, Shubing Chen, but I can't help it. She's putting out work that is, reaching great heights every other week. It's a major story, and this is no different. Story in Nature Cell Biology about Shubin's bread and butter. We're looking at disease tropisms. We're also looking at drug screening. In this case, talking about SARS-CoV-2, which we all know primarily infects the respiratory tract, but there's a wide range of disease manifestations in the gastrointestinal, cardiovascular, neurological systems. So we know it's all over the place, right? It infects multiple cell types, and organoid models have been applied across the board by many individuals. I think, Arun, you kind of dabbled, maybe not organoids, but you looked at CoV-2 and these pluripotent stem cell systems, and all kinds of different organoid systems: alveolar, lung, airway, small intestine, colon, brain, choroid plexus, heart, liver, pancreas, kidney, blood vessels, tonsils, Arun. I mean, all the organs, pretty much every single one, have been used to study viral tropism, also used for drug screens. But while there have been a lot of insights into the tropism element, many key aspects of infection remain undetermined. In particular, it's unknown whether there's common factors that you could block, if there's a molecular mechanism to block or resist SARS-CoV-2 infection across all these different cell types that are targeted. Because that would be amazing, right? If you could, have a pharmacological approach to just mitigate the damage. And that's what Shubing did in this story. She looked at changes in transcript profiles in the context of SARS-CoV-2 infection. Here's the key. They used different MOIs, and they used 3 different organoid models: lung, airway, lung alveolar, and cardiomyocytes. And they identified several genes and settled on CIART, which is the circadian associated repressor of transcription. And they showed here that if you knock out CIART in organoids compared to the isogenic control, the stem cells are significantly resistant to SARS-CoV-2 infection, even when you had viral entry. Then they did some other omics analysis to show that in part that CIART by way of NR4A1 is what mediates infection, and then moved on to look at profiling and the drug screening, which is Shubing's bread and butter, to show that the retinoid X receptor pathway is what regulates that infection downstream of CIART and NR4A1. So pretty much straightforward, although very elegant and complicated study with a lot of moving parts and some CRISPR and gene targeting and drug screening, phenotypic analysis, all kinds of omics, cut and run, ATAC-seq, RNA-seq, a bevy of assays to get at this one single point. Is there a means to inhibit SARS-CoV-2 infection across the board? And we've arrived here at a pharmacological approach, which for me, yeah, granted, we're out of the woods with COVID-2, but I think that this is all now a trial run for the next great pandemic. So I think these studies are still very important. Arun, what do you think?
Yeah, I think that Shubing Chen has been proliferative as an understatement when it comes to her COVID-19 studies. And I remember she put out some of the first organoid models of SARS-CoV-2 infection. And again, shout out to Shubing for actually citing my paper in this particular paper as well. Big plus there. But she's been so helpful. And I think really, in my mind, the scientist that I think of in the stem cell field who has perhaps had the biggest contribution to understanding the mechanisms of SARS-CoV-2 infection across different cell types and tissue types. I remember that initial cell stem cell paper, which came out in 2020, which characterized the infection of SARS-CoV-2 across all these different types of organoids. And it's just, I think, a reflection of just how collaborative she is as a scientist, because I don't think she has all of these approaches and these methodologies set up in her lab. And correct me if I'm wrong, but you see who's on this paper: Todd Evans and Benjamin Tanover and all these other experts in various other fields that are all coming together in the context of this paper. And again, reflecting on how science should be, I think Shubing is a perfect example of how science should be. A collaborative scientist, not willing to, to settle, and is always willing to step outside of her comfort area because, as of a couple years ago, she wasn't working on coronavirus. She just so rapidly transitioned into this field and achieved such great success. So I'm a big fan. Shout out to Shubing. I actually met her in person for the first time a couple months ago at our Cell Symposia conference here at Cedars-Sinai, and can't wait to see, her next big SARS-CoV-2 paper. But perhaps this is the tail end, right, as you were alluding to. So hopefully.
I don't know, man. Wait a couple weeks. You might have another one hit the rack. But yeah, as you said, she's being very nimble. And what you got to remember about her, she came out of the Mountain Lab. So you probably figure her for, pancreas, diabetes. And that is a major interest of hers. But why she's so nimble is because she really has focused on and expanded the potential of these drug screening platforms and combined it with all the other developing CRISPR, other techniques. And as she said on the show, her interests wander. She follows the question, and it leads to a lot of collaboration and to all of our benefits, because she's really been an amazing contributor, not just to this, but Zika and many things across the board. Shubin has been one of the most productive scientists, I think, objectively in the field over the last decade. So, congratulations again to you.
And she's just a nice person. That's always the most important thing. She's just a good person. All right. So moving on to a JCI paper, which will be the last paper for the roundup this time. This is coming from the lab of Laura Wood. The title of this paper is a morphology-guided transcriptomic analysis of human pancreatic cancer organoids reveals microenvironmental signals that enhance invasion. You mentioned that Shubing trained in Doug Melton's lab, who is, of course, an expert in all things pancreas. This is more of a pancreatic cancer story, but in the same realm, I suppose. So pancreatic ductal adenocarcinoma, pancreatic cancer in general, and we've talked about this a lot, is just such a sad and severe disease, and the prognosis is really grim. This is, PDAC in particular, pancreatic ductal adenocarcinoma, has major issues with metastasis, a really bad prognosis for this particular type of cancer. But what makes it so invasive and what makes this metastasis so common, we still need to figure this out. We still need to get to the roots of the molecular programs in PDAC cells that are actually driving the invasion, right? And what they did here in the lab of Laura Wood, they used a pipeline that actually used PDAC organoid isolation from patients with pancreatic ductal adenocarcinoma, and basically collected different organoids from these patients, characterized them based on how invasive they were, and then did a really deep transcriptomic analysis of the invasion profiles in these organoids. They identified differentially expressed genes in the invasive organoids compared to matched non-invasive organoids, actually from the same patients, from like a different portion of the pancreas, I believe, maybe a portion of the pancreas that didn't have the tumor or something. And they've identified 3 different transcriptomic groups in the invasive organoids, 2 of which are actually directly correlated with the invasion patterns. And some of the videos here are actually really cool. Dr. Wood actually presented on Twitter a nice Twitter thread that laid out some of the videos from this paper showing the invasion of the different pancreatic organoids into the other organoids. So check out that. Check out those videos as well. I just love papers that have videos that are tweetable. I'm a fan of Twitter, right? And that really convey the point. Because if a picture says 1,000 words, then video says at least 1 billion, right? So anyways, they did a bunch of single-cell RNA sequencing data, mapped their transcriptomic groups onto human PDAC tissue samples that are actually from publicly available data sets, and actually identified that that the critical driver of invasion here is the tumor microenvironment, and potentially even the non-cancerous cells in the tumor microenvironment can modulate the tumor cell invasion. And to further address that possibility, they performed computational ligand receptor analysis and figured out that some pretty common and well-known pathways, including TGF-beta signaling pathway, IL-6 signaling, CXCL12, are important for conferring this invasion and gene expression phenotype in these invasive cells in these pancreatic ductal adenocarcinoma organoids. All right, so ultimately they're identifying some important molecular programs that could be driving some of these invasion patterns in PDAC. And I've said this before, I'll say it again, anything that helps us understand PDAC, pancreatic ductal adenocarcinoma, pancreatic cancer in general is severely needed. And this study is an important one, very important one. It got into JCI for a reason. It's such a sad prognosis, and I think we still have a lot of work to do to understand the mechanisms of this really severe cancer.
Yeah, major unmet need. And when I see papers like this, I just think of like all the work that's been done for 100 years in 2D cell culture. Now we got to do it all over again with the organoids, because I think there's greater insight and there's more to explore. Clearly, in this case, the whole invasive angle is rendered in a much more clear and compelling way in 3D than it is, with the whole invasion in 2D culture, where the scratch and the movement and all that stuff. It just doesn't really get your socks going up and down. But seeing these images and putting it into context, I think is really impressive and shows that there's so much left to learn about this deadly disease, and a lot more ways maybe that we can get at it.
As somebody who worked in a cancer lab back in my days at Duke, go Blue Devils. Yeah, I wasn't the biggest fan of the scratch assay, I got to tell you. I spent many a weekend doing some scratch assays and migrating endothelial cells and different cancer cells, breast cancer cells. I think it's a little bit more fun. If I was an undergrad working with organoids, well, I mean, ultimately I did still go to grad school, but I think it would just be a little bit more exciting, that 3-dimensional aspect, don't you think?
Oh, for sure. And, as a segue there, because you mentioned it, this is it. Duke's playing today as we record, guys. And if they are out by the time you hear this, if you're listening, like most of you are, as soon as it hits the air, we probably won't be able to come out with the show in the next couple of weeks. They're so viciously depressed. But moving back to the show, we've got an interview to get to with Dr. Ono. Before we get there, I got a quick message from STEMCELL Technologies. Do you work with skeletal muscle progenitor cells? The STEMCELL Technologies human myocult workflow supports your muscle research from start to finish, allowing you to derive, expand, and differentiate human skeletal muscle progenitors. You can also expand mouse myogenic progenitors using the mouse myocult expansion medium. Learn more at www.stemcell.com/myocult. All right, everybody, with us today, we have a special guest from the University of Texas Health Sciences Center in Houston, Dr. Noriaki Ono, who is associate professor there in the Department of Dentistry. Dr. Noriaki Ono's laboratory studies the function of skeletal stem cells in development, disease, and regeneration of bone and cartilage through advanced applications of genetically engineered mice. Their current 3 major foci are skeletal stem cells in the growth plate, bone marrow, and craniofacial structures and molecular mechanisms governing their behaviors. Dr. Ono, thank you so much for joining us today.
Welcome. I'm really happy to be here.
Yeah, thanks for being here. Your lab is a unique operation. It's led by 2 PIs, and your lab actually intersects stem cell biology with bone and tooth biology. This is something that's not always at the forefront of stem cell biology, unless talking about like hematopoietic stem cells originating from bone marrow. But bone is actually an amazing model system for studying development. And we've talked with other bone stem cell biologists here on the show, like Dr. Joy Wu from Stanford, about how biomedical engineering, for example, is really rapidly intersecting with bone research and stem cell biology as well. Maybe one day make engineered bone tissues from stem cell derived bone one day. So in other words, there's a lot going on in this field that stem cell biologists might not know about. So I guess to start off, why don't you give us an overview of what your lab is working on these days, both on the developmental and translational side of bone biology?
Sure. So that, as you mentioned that, the bone stem cells, the skeletal stem cells, sort of kind of, second class, considered to be a second class stem cells. People, really don't focus so much effort on studying stem cells. But I guess this field is now blooming. Many people coming to be interested in this really sort of enigmatic stem cells and one of the really important feature of these stem cells is that there are many, many skeletal stem cells in many different types of tissues, and each of which has sort of different properties. So it's a really interesting, but it's a kind of really exhaustive endeavor to hunt down and try to find a stem cell that can be used for things like tissue engineering or things like that. Right. So what we are trying to do is, our lab is by no means any sort of, IPS, lab or things like that. My background is in bone developmental biology, and we have interested in how bone stem cells, what kind of cells do they become. So that we are primarily working on this technique called in vivo lineage tracing techniques, so that we can mark a specific bone stem cell population in vivo. And so then we can just follow their fate over time using, imaging or, histological sections and try to find out where they go and what they do. And we also combine these techniques with a bunch of other standard stem cell techniques, such as, colony forming unit fibroblast assays and single cell RNA-seq or things like that. So the fundamental question we are trying to ask in the lab is that because as I mentioned, because there are so many different subtypes of skeletal stem cells in different part of bones, like, the bone at the edge, the bone in the center, on the tooth, or in the craniofacial bone, the skull suture. So we're trying to find, trying to hunt down many different type of skeletal stem cells in different locations. And if there's anything that is common, or they, sharing some characteristics. And also we are also interested in translating these stem cells for regenerative purposes. So we're trying to, in order to do that, we have to know what kind of molecular properties these stem cells have. So that's also things that we are trying to do. But we are not a very translational scientist, so we are just only trying to generate a fundamental knowledge about these properties of stem cells in many different types of bone tissues. So that's what we are doing.
Yeah, maybe not explicitly translational, but I think there's a lot of implications for the translation of stem cells and generally, but also specifically in the bone to your work. And to that point, while bone disease maybe not the most publicized target of cell-based and regenerative therapies, the use of, I guess, stem cell adjacent, you could call them, approaches is widespread in orthopedics, mostly for elite athletes, right? Whether it's this platelet-rich plasma injections or microfracture treatments and other treatments of questionable efficacy, maybe pseudoscience there in some of them, but there's a lot out there for elite athletes. And then there are these genetic, developmental, and degenerative diseases that are less widespread, but much more devastating. For example, osteogenesis imperfecta, osteoporosis, bone cancer. So there's a lot of targets, I think, not just in sports, but more, I'd say, in medicine and disease. Is there, like, I mean, I'm betraying my relatively superficial understanding of bone stem cells, but is there a universal cell type that would or could address all of these conditions? Or does each of these conditions require tailored cell type and or approach?
Yeah, I guess that's a really important question. I don't think we know any, definitive answers to your questions. But I guess that, bone stem cells, skeletal stem cells have really sort of wonderful capabilities of course, these are stem cells. They can be, almost indefinitely, be expandable, and especially when they are isolated in vitro. So then, and the other really interesting property is that they secrete a lot of cytokines and sort of, tropic factors to help other cells, maybe doing some good things or bad things. So I think there are two important characteristics is that number one, they can expand a lot. So, in taking advantage of that, capacity, people use these so-called mesenchymal stem cells injecting into some sort of injuries or, some degenerative conditions and, try to improve that, the condition. I think that's, it's not a really sort of authentic application of stem cells, right? It's not like stem cells themselves, repairing the tissue. It's just the factors that are released from these stem cells that are helping the endogenous tissues to heal. I think that's one clever application, but it's there are many efficacy issues and consistency issue, right? The reproducibility issue. So then I think that kind of the quality control is a huge issue in the field. And of course that in, let's say, huge bone defect or like, sort of unhealed fractures, large unhealed fractures, of course there are ways to, ask these stem cells to become bones. Maybe we can just need to get some help from tissue engineers to facilitate these cells into bone cells. So I think it's there are two aspects, right? So I guess that I think that the field is still trying to learn that what is the best way of, how can we take advantage of these stem cells? And of course, my interest is that, how we can assist or, enhance these endogenous naive stem cells, to do a better job. But there are many different aspects. And, it's there's so many investigators who are interested in, taking advantage of these amazing capabilities.
Yeah, it's such a powerful cell type, cell lineage. And then there's, like you just said it yourself, there's so much that we don't know about how the bone develops and how cartilage develops, for example, and how these different cell types and these niches crosstalk and interact with each other. And this is actually something that you studied very recently in a recent paper, a Nature Communications paper, that actually dove a little deeper into this developmental crosstalk in the bone, examining the fate of early perichondrial cells and developing bones. And this study actually used a lot of the in vivo cell lineage tracing technologies that you're alluding to actually show that these fetal perichondrial cells contribute to bone and marrow stromal compartments under the regulation of a pretty famous signaling pathway, hedgehog signaling in particular. So take us through this work that came out recently and kind of more broadly talk about some of the, in your mind, the biggest remaining questions in understanding bone development.
Yeah, so then I guess that we are all, also, trying to understand it, but there are many different types of stem cells that I mentioned. And it seems that the property changes over time. So, the stem cells in age time and the stem cell in young time, they're different. So, I think one of the most sort of translationally relevant question is that can we actually, rejuvenate, all stem cells to young ones to facilitate healing? I think that's one of the very exciting pathway that need to be sort of addressed, and we are interested in try to identify the signatures of these old stem cells versus young stem cells and how they can be convertible. And the other thing is that we really want to know is that what is the role of these skeletal stem cells in these famous bone disease conditions such as osteoporosis, or there are bone cancers or things like that. I guess there are multiple open-ended questions in the field. But I guess that still right now the biggest limitation is that yes, we know that these skeletal stem cells can be isolated in vitro and it can be transplanted, but where these cells are actually located in vivo and what they are doing in naive condition, I think that's also really still remaining. People now trying to understand more and more, but it's still insufficient. So there are 3 questions that we're trying to ask right now in our lab.
All right, so forgive me, this may be a bit outside your expertise, but I'm sure you've considered, I mean, your focus on the niche, and you alluded there earlier to the trophic factors that are produced by these skeletal stem cells and their derivatives. So I'm sure you have very much considered the niche and the milieu and secreted factors there, and perhaps being that you're focused on this fetal bone marrow, or what will become the bone marrow, you may have considered the impact your work could have on the hematopoietic stem cell field. Arun will tell you, I am a real fool for the blood, and I always try to bring the conversation in that direction. So forgive me, but the niche that fosters hematopoietic stem cell genesis and maintenance is like a holy grail in hematology and more widely in translational medicine. And the components of that niche have stirred fierce debate. Is it the endothelial cells, periosteum, mesenchymal cells, leptin-positive cells? There's a case to be made for all of them, and I'm sure that they're not mutually exclusive, but your work, it's focused on the endochondral bone development, and that involving the bone and presumably the marrow. Do you have any thoughts about the hematopoietic stem cell niche specifically? Do you think that these fetal cells, and this is where my interest really is, because this is like a pluripotent stem cell show, do you think these fetal cells that you've identified could contribute to that early niche that supports hematopoietic stem cell establishment, colonization and establishment in the marrow?
Yeah, definitely. I guess that's one of the most sort of prime example of how development of the biology can help let's say IPS researchers to identify the factors to recruit and retain in the bone marrow. I guess what I can tell is that bone establishment of bone marrow hematopoiesis is very, very organized, and it's a really, really important step. So I guess one of the very critical the event for that to happen, for the hematopoietic stem cells to come to live in the bone marrow, is that these chondrocytes in endochondral bone development, these chondrocytes have to undergo hypertrophy and become a very specialized sort of ballooned up cells. And they, some of them, these cells die, but some of these hypertrophic chondrocytes somehow can survive and transform into marrow stromal cells. And we know that these hypertrophic chondrocytes express a lot of trophic factors, including VGF and Indian hedgehog and whole different types of cells, the trophic factors. So I think that there is something really sort of magical about these hypertrophic chondrocytes, that they either die or survive and become bone marrow stromal cells that support bone marrow hematopoiesis. So I think that is going back to your original question, that thinking about the niche for hematopoietic stem cells, I think that it's really important to think about the mechanism and what kind of transition is going on between these hypertrophic chondrocytes, eventually becoming into marrow stromal cells that support hematopoiesis. So I think there will be a lot of things we can learn from these type of cells. Now, having said that, there's no really good assays to understand the biology of hypertrophic chondrocytes at this moment, because hypertrophic chondrocytes is so big. It's about 100 micrometer diameter, and it's such a fragile cells. So that let's say conventional single cell RNA-seq analysis, they cannot capture these cells. We are all missing out these cells. So that we have to have some sort of more advanced, let's say, spatial, but transcriptomics or things like that to understand what's going on in these cells. But I think that, and of course, as I recently we showed that these perichondrial cells also contribute to the formation of the niche. So it's a really complex process. Not only hepatochondrocytes, but also perichondrial cells moving into the marrow cavity with blood vessels and make some sort of really magical place for hematopoietic cells, stem cells coming into migrate into the bone marrow space. I think that's a really interesting question. I don't know what is the cocktail or the mix that welcome hematopoietic stem cell to the marrow space, but I guess that's a really, really important thing that we can learn from bone developmental biology. Yeah, that's still largely unresolved.
Yeah, and like you alluded to, I think there's a lot of really exciting technologies that'll help us answer some of these questions. And I think, it's a given that this is the Stem Cell Podcast. And of course, on every episode, Dalen's going to bring up the hematopoietic stem cell in some capacity. This is a running gag for us. So thank you for answering that question. But, we're talking about bone biology here. And there's another aspect of your lab that we alluded to, which of course is the focus on tooth development and tooth biology. And it's not often that we talk about the intersection of dentistry and tooth biology with stem cell biology, but both you and the co-PI of your lab, Dr. Juanita Ono, are dentists by training. And in fact, just looking at the composition of your lab through your lab website, it seems like most of your trainees actually have a dental background, with many of your lab members having trained as dentists in Japan in particular. It's such a unique path, and it's not something we talk about a lot here on the show. So I'm just really curious about your training and your path as a dentist / stem cell biologist. And also, what inspired you to go into the stem cell side of things as a trained dentist? So tell us more about your path.
Yeah, I mean, thank you for asking that. So, one of the things that I have to tell you is that, my family is a heavy dentist family. So my mom, my grandpa, I mean, I'm a third generation dentist. So then, it's a long history. But I mean, when I, went into dental school, it's like, dentistry is more about the art, but, there are some other science component, but it's also pretty much, it's sort of different things, the art and the science are pretty much different. I was really fortunate that when I was in dental school, there are many, dentist scientists, researchers who have contributed substantially to the biology of bone and tooth development. So I was kind of really lucky enough to be exposed to the excitement of science, especially regarding bone biology. And when I did my PhD, I was really lucky enough to land on a lab, who is a prominent biologist, prominent bone biologist, Dr. Masaki Noda. He's a very prominent bone biologist. So then my training during my PhD sort of expanded my perspective. Not only, it's not, the dentistry is not only about, tooth. It's like there's something else that we really, really, need to care about. So then, that was around the early 2000, and that was the time that, sort of the stem cell biology started to explode in the field. So through reading all these literatures and one wonderful story, especially about actually hematopoietic stem cells, right? Skin, intestinal stem cells, skin stem cells. Then, I thought about maybe, we have to learn more about stem cells in bones and teeth. So that's how that inspiration that I got, around 20 years ago when I was a PhD student, it just continued on and on and to this day. So I was lucky enough to be supported by excellent mentors who supported my journey to here. So it's and of course the other thing I'm really pretty much dedicated to sort of fostering, because I'm a dentist myself. So I really want to foster the next generation of dentist scientists, because it's really important for it's a very small cohort of scientists, right? So I think it's I'm a unique position to sort of help them to find more exciting projects and exciting directions that they can carry forward. So that is sort of my dedication and my commitment. So that's why, and also, because I have a connection to my original alma mater, then it's relatively easy for me to find highly motivated individuals who are interested in doing this skeletal stem cell research. So so that's how I came along. And yes, I think I know I have a really unique background, but actually it really helps me to understand the whole picture. I'm an orthodontist in training, and in orthodontics, really deals with the bone development in the skull and in the face. So we really understand the value of how significant is the bone development biology / stem cell biology can be in the clinical relevant field. Yeah.
Yeah, you are a rare breed, Noriaki, and not just a dentist in stem cells, but a dentist in stem cells who appreciates the beauty of the hematopoietic stem cell. We need more of you, my friend. But moving back to the bone, as Arun is probably going to yell at me to do, we're going to get back to the bone. It's well documented that bone repair and homeostasis in adults is governed in large part by mechanical forces. And yeah, that totally makes sense. And biomechanical influences are increasingly believed to play a role in a host of stem cell differentiation processes. There's this variable stiffness of the growth substrate can have a modulatory influence on differentiation of mesenchymal stem cells. That was published like over 10 years ago, and we made a big splash, but also shear stress shown to play a role in hematopoietic and vascular differentiation and other components of the mechanical environment, from hydrostatic pressure to even microgravity. We could get Arun to talk about that. Have also been implicated in stem cell differentiation. But in the case of fetal perichondrial cells, perhaps there's not a lot of mechanical stress there, floating around in the amniotic fluid. But you've also studied unique skeletal stem cells in the growth plate in adult mice. So you, I'm sure, are familiar with this notion. Have you considered the potential role of mechanical force in driving unique properties of differentiation in either that fetal perichondrial context or in the growth plate? What do you know about that?
Yeah, it's a really fantastic question. So that there is no doubt that mechanical loading is an essential component of forming probably the niche for skeletal stem cells. And one of the really interesting findings that we came across when we identified the skeletal stem cells in the resting zone of the postnatal growth plate. So these stem cells are formed in the specific portion of the growth plate called the resting zone. But that the formation of that layer actually coincides with the formation of this, the supporting niche called secondary secretion center. And that structure, occurs only postnatally, when the baby mice start to crawl and things like that. So I think that there's a lot of correlation and probably causative relationship, which is that the mechanical loading on this cartilaginous structure will sort of induce cell differentiation and a vascularization that makes a new environment a niche, and that in turn switch on another niche factor and stimulates to form another chondrocyte, the growth-based stem cells. So I think a really important there's feedback loop, and it's mechanical loading is definitely one of the most important factors. And we know that mechanical unloading leads to bone loss, and which is of course in part mediated by bone resolving osteoclasts, but also it's for sure that the cells are sensing this mechanical loading are the cells that are on the bone surface or within the bone. So within the bone, there are osteocytes, they are, projecting like, almost like neuron, they're projecting these dendrites, connecting each other and they're, share stress and food stress, going around. And these cells, sensing this, alteration in mechanical environment and changing the signal, on the bone surface. And we think that there are some, these skeletal stem cells near the bone surface. So there are cells probably directly or indirectly sense these changes in mechanical loading and, sort of, start to change the behavior. So it's a whole cascade of direct and indirect, the thing. And, mechanical unloading, it's one of another really mysterious factors. And it's depending on the direction of the force and the side of the force exerted, you have differential reactions. So it's not like, side kind that is, it can be explained by gradient. It's like, it's a more three-dimensional, more complex things. But considering the skeletal stem cells are very meticulously anchored to the extracellular matrix, mechanical loading is going to be one of the, next important factor that need to be sort of disentangled, what they do in stem cell self-renewal and cell differentiation. It's a really, really deep and, never-ending area, I think. Yeah, it's a really important.
Yeah, it's a very hot area of study. I mean, of course, the Nobel Prize a couple years ago was given to mechanobiology and mechanosensing and the piezo channels as well. And certainly in the stem cell field, we've alluded to just how important mechanosensing and mechanobiology is to differentiation, all different aspects of stem cell biology, really. And we also talked about the importance of the niche a lot here on this conversation. But I think in addition to the importance of the niche and the biology, there's also the importance of the niche in the development of a biologist, so development of a scientist, right? And you mentioned yourself, you have such gratitude to your mentors for developing you and providing you the right niche to become that, dentist, stem cell biologist that you are. And you actually recently moved your niche. You moved to the University of Texas Health Sciences Center at Houston. That was my transition there.
Yeah.
Hope you enjoy that one.
Yeah.
Yeah. So you recently moved to the University of Texas Health Science Center in Houston after being at the University of Michigan for a while, a few years. And some folks actually may not know this, but UT Health is located in the Texas Medical Center, which is actually the biggest medical center in the world. It's a ridiculously gigantic complex, which kind of just blows my mind about how many different institutions they are in just that square mile area in Houston. I think there's more than like 60 medical institutions in this 2 square mile area of Houston, and some real powerhouses in medical research in this area, including the Baylor Medical Center, Houston Methodist, MD Anderson, and I can go on for forever here. The reputation of the medical center really speaks for itself. But, tell us about that transition. Tell us about your time so far at UT Health and what made you move there, and, what it's like to work and collaborate in such an incredible place for medical research.
Yeah, thank you for asking that. I mean, sort of as a academic researcher, it's moving across the country is also a process. So meaning that I've been in the United States for 14 years already, and this is my third institution. So it's I think it's a real process for me. It's really by moving around different institutions, I feel that I'm growing up a lot, because getting to know different systems and different types of regulations or protocols. So it's really a process. So and it's I did my postdoc in MGH under Hank Cronenberg, and he's a prominent endocrinologist. And it's MGH is a fantastic institute, and it's a really also very competitive institution. And I got all the scientific inspiration from there. And of course, then I moved to Michigan. Michigan is also top notch, really fantastic institution. And so then it's I kind of really being kind of continue to be exposed to different institutions, different really nice institutions. So I'm also, again, have to say I'm really lucky enough to be in the really top notch institution. And of course, I know the reason that I moved down to Houston from Michigan is that it's being assistant professor. I was an assistant professor in Michigan, and then I moved out to a tenured associate professor position. But it's a promotion and a tenure is just only sort of one thing, right? It's only one sort of award and it's a recognition. But at the same time, I was also looking for the opportunity to expand and continue the momentum of my research. So then, really, all about the timing. That and it was lucky but unlucky that it was a COVID time, so my lab didn't have so many people in my lab, and my lab was relatively compact at the time before moving here. So I just took the opportunity to and just make another move from Michigan to here, and it's really a fantastic environment. And there are so many different institutions. So then I think it's by moving around across the country, it makes me get to know a lot more people by each move, right? So it's really really an asset for me. So it's really all about me growing up and me getting to know all the different types of people, and me sort of expanding my network. So but it's not easy actually. Moving across the country with my spousal investigator, Dr. Juanita Ono, and my kid. It's not easy, but the reward is actually pretty high. And I was pretty much sure that coming here, my research program will even grow bigger. So that was what I was aiming for. But that's a really one of the most difficult decisions that we have to make in our the career as scientists. But yeah, I've been pretty much lucky that I have identified really nice opportunities each time.
So I want to circle back to your family history a bit. I mean, you were born to be a dentist slash orthodontist, right? And you kind of went your own way. We talked about that, but you're talking to a guy here who's had braces 3 times. Okay, I've done it all. I had the regular, I had the lingual, I had the Invisalign 3 times in my life. When I was a teenager, when I was in grad school, and just recently a couple of years ago. And what I'll say about orthodonty, if that's how you say it, is that it's really advanced, the technical elements of it, right? Like back in the day, they put a whole mass of clay in my mouth to get the molds. Last time I went, they just put the wand in there. They do like LiDAR pretty much to get the scan. So, I mean, I think that's for me just a vivid example in how the clinical or health care providing landscape has really changed due to technology. So I want you to paint a picture for me. I mean, I look at your papers. You got the most beautiful images, and talking to an expert in any field is a great opportunity. And as you've done already on the show today, you've kind of sketched out what the niche looks like and the different cell types. So I want you to paint a picture for me. Maybe you will never step foot into the clinic again, but do you envision that there will be an integration of like cells with your garden variety dentistry, orthodonty? Or are we just talking about models for understanding, craniofacial defects and or bone disease? Is there really at some point going to be a cell-based dentistry, do you think, or is that just unnecessary at this point?
Yeah, I think it's actually happening. I mean, meaning that, by the way, I haven't really renounced my status as orthodontist. I'm trying to be engaged in graduate orthodontic, the program. It's like, and I go to the clinic and just stand there and, guide residents, only twice a month, and that's it. And that's enough for me. But, I think it's really important for me to keep that aspect. Anyway, I think that the cell-based, dentistry, so that actually orthodontics doesn't require so much cell-based therapy. However, there are other specialty like periodontics. There are many patients who lost the periodontal bone, then they need to regenerate bone. In that kind of case, there are many applications. There are many opportunities for regeneration, such as that there are many clinicians use some sort of trophic factors, right? BMP2s or some of the factors to apply these factors into the defect so that they can make more bones, so that they can do the restorative procedures, much more effectively. So there is the dentistry is, of course, at a much smaller scale than, let's say, orthopedic reconstructions and things like that. But I guess there is there's a lot of opportunity for regenerative medicine. So I think that I have to tell you that regenerative medicine is a big agenda in dentistry. So that I think it's almost like, as the technology for stem cell-based therapy become more sophisticated, improved, suddenly there will be, more sort of unique and, probably new application of this technology to regenerate, the bones and tooth and their surrounding tissues. So I think there's no doubt it's deeply relevant to dentistry. Yeah, that's something I can say.
Well, great. I mean, maybe my fourth time around with the braces won't need any cell-based therapies, but by the time my teeth start falling out, I guess I'll have someone to talk to. Anyway, thanks for this great conversation. We're not going to let you leave just yet. We got a couple peripheral questions, but it's been so great talking to somebody in a field we don't talk about enough, which is, the teeth and the mouth and the craniofacial and bone, all of it. But before we let you go, we have a couple science-related questions for you. First is, what is one hobby that you always wanted to pursue but were never able to?
Yeah, actually, I really, wanted to play the piano, but I somehow, missed the opportunity to do that in my career. And, when I, grew up, I never had enough time to do that. Actually, my 7 years old, she's doing the lesson. So I'm kind of, sitting behind and trying to understand. But, I have so many other things to do. So I don't have time to do. So maybe after I retire, maybe I have to pick up this hobby and try to spend time to learn the lesson. And what I learned from my daughter's lesson is that this is very, very sort of systematic. It's almost like she's learning the way how to code in the algorithm. So this is a very, very good, fast way of sort of scientific education. So I'm really impressed that how, these, the piano is actually a very making a good foundation for, think about anything in a very systematic way. So this is something I really want to do, but I haven't done that. And I hope that I'm going to be able to do that after retirement.
Don't wait. Hey, I mean, it's never too late. Everyone talks about the plasticity of a child's mind and learning a musical instrument, but just take inspiration from my co-host, Arun, who has picked up the guitar and started strumming. And, with you on board and a few other guests that we've spoken to who have aspirations in a similar vein, I think we've got the critical threshold to start our stem cell jam band. I will be singing lead vocals. Apologies in advance to anybody who has to hear it. Finally, the last question. What is the best piece of advice that you've ever been given, either professional or not?
Yeah. So, I have to admit that I have received so many good advices from my mentors and my friends and my colleagues. But by far the most impactful one was that the one from my mentor, he said that this can be a little bit, cruel, but he told me that I find my own way and you find your own way, which means that, you got to find, what you want, and don't, rely on me too much. You have your own passion, of course, you have your own career. And so that's the most impactful word. It's it's all about the context, right? But in that specific context of conversation, this was a hugely, helpful for me to find my own way. That's why I sort of it's just simple like 6 words sentence can have huge impact for somebody's career. So I was really amazed at how impactful this advice can be. Yeah. So I'm still finding my own way. Yeah.
Well, I think you've zeroed in on it. You've found a good niche, so to speak, and that is great advice. I mean, no scientist wants a clone of themselves with the same ideas, and that's what really builds innovation, right? It's giving a little bit of yourself to the mentee so they can make the most out of what you couldn't achieve. And I think you are a great example of that on both sides. You've really transcended dentistry and orthodontics and gone deep into stem cells, and I'm sure you're an even better mentor to your group. So appreciate you just on behalf of all the people in your life, but also appreciate you joining us to talk to us and our listeners about your unique path. Noriaki, thank you again for joining us on the show today.
Thank you. My pleasure.
All right, everybody, that brings us to the end of this episode. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and roundup papers. You can also reach out to us on Twitter at StemCellPodcast or via email at info@stemcellpodcast.com with feedback or to suggest guests. Until the next episode, that'll do it for this couple weeks. I'm off to fill out my bracket and watch Duke struggle to win, hopefully.